Thank you for that introduction. I'm Graham Shulver, Senior Application Scientist at IDEX Health and Science, and welcome and thank you for joining us at today's webinar on IDEX Health and Science's capabilities for monoclonal antibody characterization workflows. Accurate analytical characterization of monoclonal antibodies is essential for monitoring critical quality attributes and critical process parameters in biopharmaceutical manufacturing. IDEX Health and Science, being a leading global innovator in fluidic technologies, is ideally positioned within the industry to provide engineering and manufacturing support for biopharmaceutical analytical initiatives. IDEX's product development model involves partnering with instrument manufacturers to provide novel solutions for even the most challenging bioprocess analytical workflows. Increasingly, biopharmaceuticals in their many forms are positively affecting patient outcomes. However, developing and manufacturing these complex products provides several analytical challenges. Streamlining the analysis of biopharmaceuticals through automation can significantly improve insight into production processes, which in turn can accelerate product development timelines. Being the market leader in life science fluidics with extensive application and core technology expertise in sampling, sample preparation, chromatography, and mass spectrometry, IDEX is the perfect partner for instrument vendors supporting bioprocess PAT. IDEX's unique product development model pairs our fluidics design, development, and manufacturing teams with instrument vendors' hardware engineering groups. This combination results in superior analytical tools that enable the production of game-changing biopharmaceuticals. At IDEX, we are focused on bridging the gap that exists between monoclonal antibody production and the analytical instrumentation required for monitoring these processes. The lack of real-time critical quality attribute and critical process parameter data from bioreactors has long been identified as an industry pain point that adds unwanted time and complexity to drug development and manufacturing. IDEX is actively applying its expertise to develop tailored solutions for online and at-line analyses being used to characterize therapeutic biomolecules. Some of the core fluidic functions being used for monoclonal antibody characterization include sampling, filtering, purifying, diluting, reacting, separating, detecting, and many more. At IDEX, we provide instrument vendors with engineering and application solutions that directly benefit their customers in terms of ease of use, time to market, and technology differentiation. I'd like to thank you for your attention. And with that, I'll hand over to my colleague, Craig Love, who will be highlighting several examples of fluidic workflows specifically designed by IDEX for monoclonal antibody characterization. Over to you, Craig.
Thank you, Graham, for the introduction and for reviewing some of the core technology highlights for IDEX Health and Science. At this point, I would like to provide some examples of IDEX fluidic capabilities that can be used to address specific challenges in monoclonal antibody characterization workflows. The first fluidic system I would like to present is for automating antibody calibration. Creating a calibration curve is the first step in determining the antibody concentration in a sample, which is generally referred to as antibody quantification, or titer. Accurate measurement of antibody titers is essential for process modeling, monitoring, and for optimizing process control. Let's take a look at how IDEX technology can simplify and automate the antibody calibration process. Generating antibody calibration curves is typically a laborious process that requires preparing multiple serial dilution levels. In addition to being time-intensive, this process is also prone to dispensing errors in sample and diluent volumes. This can cause calibration outliers that affect slope and intercept values, or result in lost time preparing and running new calibration standards. The IDEX approach simplifies the calibration process by using a single calibration standard together with a highly accurate dispense pump to load variable standard volumes onto an affinity column, such as protein A. Upon elution, the antibody peak can be measured using a compact UV LED-based detection system. Using variable injection volumes eliminates the need to prepare serial dilutions, which saves time and reduces the likelihood of dilution errors. This approach also allows the calibration subsystem to operate without an autosampler for simplified control. Another benefit of the IDEX approach is flexibility. The simplified schematic shows only a basic calibration configuration, but additional fluidic connections can be added to provide sample introduction for automated titer measurements, multiple calibration standards, or additional buffers for sample wash, column regeneration, or clean-in-place protocols. Standardized fluidic connections make it easy to interface with other types of optical detection, such as fluorescence or Raman. Lastly, the small footprint and industry-standard I2C communication simplify integration of fluidic subsystems into new hardware design projects. Another important analytical workflow for increasing antibody yields during production is spent media analysis. Media optimization not only improves cell health and viability for improved titers, it can also improve product quality by limiting undesirable metabolic processes. Spent media analysis is important for process monitoring and modeling to ensure ideal growth for each cell line or clone. Although several analytical approaches may be utilized for spent media analysis, one of the key methodologies is UHPLC-MS-MS. A typical sample preparation workflow for spent media analysis involves protein precipitation with acetonitrile, followed by centrifugation, filtration, and finally, dilution. Because this is a quantitative workflow, the consistency and accuracy of each step is essential. With a manual workflow, variability can be encountered in each of these key process steps. On the next slide, we will discuss how IDEX technology can streamline sample preparation for spent media analysis. The IDEX approach results in highly reproducible sample preparation by eliminating the protein precipitation and centrifugation steps for improved process control. As shown in the patent-pending fluidic example, a sample aliquot is first loaded into the sample loop of a specialized injection and mixing valve. The valve is then moved to the inject position, where one pump is used to elute the sample loop into a diluent flow, which is controlled by a second pump. The dilution ratio is determined by adjusting diluent flow relative to the sample flow. Using this approach, dilution ratios ranging from 10x to more than 100x are readily attainable. The next step in the IDEX approach is to replace the laborious precipitation and centrifugation steps with a replaceable ultrafiltration module. This approach separates large protein molecules from the cell culture media components without the addition of organic solvents such as acetonitrile. It also performs effective sample filtration, so no additional process steps are required. In addition to improved reproducibility, another benefit is the ability to capture all of the process parameters, such as flow rates, calculated dilution ratio, and ultrafiltration module pressure in electronic format. This allows for more effective process control, tracking, and troubleshooting. Attack mass analysis of monoclonal antibodies typically utilizes high-resolution accurate mass, or HRAM LC-MS instrumentation, to measure the molecular weight of intact antibodies, either in native form or under denaturing conditions. Prior to electrospray analysis, the antibody must be purified to remove salts, detergents, and other proteins that may interfere or complicate analysis. Conventional workflows often utilize ultrafiltration, typically in the form of centrifuge spin tubes or protein precipitation, utilizing organic solvents to purify antibodies from complex solutions, such as cell culture media. These approaches require several steps and often have variable recovery percentages. What if salts, detergents, and protein interferences could be removed in one automated step? On the next slide, we will discuss how IDEX technology can streamline your workflow with smart, intact mass sample preparation. The IDEX approach to smart antibody sample preparation for intact mass analysis utilizes protein A purification with smart control of monoclonal antibody concentration. The first step in this approach is to load a sample aliquot into a holding coil or sample loop using a rotary selector valve and an IDEX dispense pump. A titer measurement is then performed using part of the sample aliquot together with inline UV LED detection at 280 nanometers. Based on the titer measurement, a known amount of the remaining sample aliquot can then be loaded on the protein A column, washed, and eluted at the target concentration using a mass spec-friendly elution buffer. The smart aspect of this approach is twofold. The first advantage is that the desalting and washing steps can be automated while avoiding cumbersome centrifugation or protein precipitation workflows. The second advantage is that the purified protein concentration can be controlled for optimal mass spectral response. This approach avoids inadequate signal response or detector saturation that can negatively impact intact mass measurement. Finally, if the product is delivered to a sample loop after purification, pH adjustment can also be performed by mixing with a neutralizing buffer prior to LC-MS analysis to optimize charge state distribution. Due to the structural complexity and inherent heterogeneity of biopharmaceuticals, such as monoclonal antibodies, the monitoring of product quality attributes, or PQAs, has typically required an array of chromatographic and electrophoretic separation techniques. More recently, a multi-attribute method, or mAb, approach has been adopted to monitor multiple post-translational modifications, or PTMs, of biotherapeutic molecules. This technique utilizes high-resolution LC-MS-MS-based peptide mapping methodologies to search for predicted peptide sequences and fragments using accurate mass spectral information. A typical triptych digest protocol for mAb sample preparation involves denaturation, reduction, alkylation, and finally, the triptych digestion of the antibody sample. A similar sequence of steps is required for released N-glycan analysis, which generally uses a pNGase-F digestion for glycan release. Automation of these complex monoclonal antibody sample preparation workflows usually requires a complex robotic liquid handling system. However, the cost and complexity of these systems is often practical only for high-throughput laboratories. IDEX technology can provide another option for low and intermediate throughput laboratories with flexible integration for analytical systems. The IDEX approach for monoclonal antibody sample preparation begins with a customized fluidic solution to meet your specific sample preparation needs, whether it's peptide mapping, subunit, or released glycan analysis. In this example, an IDEX dispense pump and rotary selection valve are configured to utilize air-segmented fluid delivery. This approach enables aspiration and dispensing of very small sample and reagent volumes with sub-microliter accuracies. It also enables the movement of fluid segments into different heating and mixing zones to support complex workflows, such as in-solution or magnetic bead-based triptych digestion. Inline sample purification options such as filtration, desalting, and magnetic bead or solid phase extraction can also be incorporated. For accurate control of process parameters, a variety of intelligent monitoring options are possible, including temperature, pressure, flow, and bubble sensors to precisely control fluid position and delivery. This simplifies direct integration with analytical instrumentation or external devices. In closing, I hope that these examples have demonstrated how IDEX fluidic capabilities can help advance your monoclonal antibody characterization workflows. With the IDEX portfolio of products and design expertise, we are ready to partner with you to develop a customized fluidic solution or integrated subsystem to meet your project needs. If you would like to learn more about partnering with IDEX Health and Science, contact one of our fluidic experts or visit our website at www.idex-hs.com. Thank you.